Good morning, everyone. Welcome to class. It is Wednesday, April 1st. No April Fool's Day for anybody today. Everybody's way too serious. Hopefully you are doing well, feeling healthy, feeling happy. We've got a busy schedule for us today, a fair amount of lecture to do. We got most of our anatomy done in class last time on Monday. So we get to start talking about the urinary system physiology, the process of making and processing our filtrate, or making and processing our blood to produce the urine as that byproduct. Again, you're supposed to be using your own time, your lab time, to work on that anatomy. Make sure you understand everything on that list, because again, your urinary and respiratory lab exam is going to be equally weighted, 50%. We've had 3 lectures on your respiratory system, 3 lectures on the urinary system, so it'll be 50-50 in both. And it should be a good size lab exam, probably somewhere close to 70 questions. To help you prepare for that, and again, by that, I mean only the process, making sure that your system is compatible with the process. I do have that practice proctored lab exam. 5 very simple, very stupid questions. Just to give you an opportunity to make sure that your camera and your microphone and everything else works. So this is a chance for you to see what is going to happen on the real exam. So you have the opportunity to make sure your system is compatible, make sure you have the microphone, make sure you have the camera and everything else that will allow you to work. This should hopefully help us to work out the kinks so that when we do the tests for real on the 15th, that we won't have any problems with that, or at least limit the problems. We do have 2 more assignments. They're due after spring break. Your Unit 26 review is due, and your PhysioX Exercise 9, which has 6 activities. And again, especially now that we are doing our labs at home, that PhysioX is a really important tool, so make sure you are doing those. As I also mentioned, excuse me, as I also mentioned last week, we don't have lectures next week during spring break, but I will be working on the exams, and so I will be on my computer a lot, so I am going to be doing office hours. I will be doing office hours for us on Monday and Wednesday from 10 to noon. I will also be doing office hours on Tuesday through Thursday from 12 to 2. I'm happy to answer any questions during any of those 4 days and any of those times. I will say that, again, any student is welcome to come to any of those times. However, on the Monday and Wednesdays, technically those are my 4:31 office hours, so you guys will have priority over any 4:30 students, but if nobody's there and a 4:30 student wants to come in, great. Same thing on Tuesday and Thursday. Technically, that is my 4:30 office hours, but if no one's there and you come in and you have questions, I'm happy to answer those. And if for whatever reason those times don't work for you, although those are during our class time, so it should, you can always email me and we can make other arrangements to meet in Zoom and discuss these materials. All leading up to Wednesday. Again, remember also that means on Monday, the 13th, we do have a full lecture. Again, it is a lot of new material 2 days before the exam, just like if we've had to do in the classroom as well. But just like in the classroom, you have the lecture notes, you have the textbook, you have the resources. So really you should be using spring break to look ahead. So Monday's lecture when we come back really should just be review. You should have looked at all that stuff during the break, prepared for it during the break. We'll review it then on Monday. And so it shouldn't be new information for you 2 days before the exam. You have a wide window to take the exams on Wednesday. It'll start at 7 a.m. And we'll go through 2 p.m. And again, you will have both the lab and the lecture exams. You can take them in any order that you want. Again, the timing for those will depend on how many questions and stuff on it. My guess is somewhere between 45 minutes and an hour for the lab exam, probably an hour because of the number of questions. The lecture exam, my guess will be somewhere between 1 and 2 hours for that. Again, depending on the number of questions. So you'll have that to go with. And then, but make sure you give yourself enough time. Again, don't start the exams at 1:45 in the afternoon because, again, they're going to close at 2. So make sure you give yourself plenty of time to answer all the questions and do the things that you need to do. All righty. Any questions on that? I guess before I started talking, I should have asked you, as always, to raise your hand or give me a thumbs up or a yes. Can everybody hear me okay? Excellent. That means I don't have to do all of that introduction again. All right, so any questions on that introduction? Anything else that we've got planned for today or coming forward for the next couple weeks before we dive back into the lecture? All righty, then away we go. Let's go ahead and switch to our, oops, why is that not working? Hold on 1 second. Oh, that's not fun. For some reason, I don't seem to be able to share my slide presentation. That is a fun and exciting new. That's weird. All right, hold on 1 second. All righty. Okay, it seems to be working the way we want it to work. Let's see if this continues. All righty. So we left off last time, and we were looking at our anatomy, as we mentioned. Can you still see that? Yep, that still works. Okay, excellent. All righty. Excuse me. We left off last time and we were talking about the anatomy. Again, what we primarily were focusing on, where the bulk of the anatomy was located, was in our nephron. That is where all the magic happens. So we have the physiology there. So this is what we wanted to emphasize. Again, remember the key to our nephron is it is made up of two components. It is made up of tubular, renal tubular structures, and it is made up of blood vessels. Here, this illustration, again, we emphasize the tubular structures. They start with the Bowman's capsule or glomerular capsule, then the proximal convoluted tubule, then our loop of Henle, and then our distal convoluted tubule. And we talked about the anatomy. As we mentioned, the glomerulus and the glomerular capsule and the space in between them collectively form our renal corpuscle. And it is that renal corpuscle where the filtrate is made, and then it is the rest of the tubule where we process that filtrate. And as we talked about, the majority of that processing occurs in the proximal convoluted tubule. So we will talk about that. The distal convoluted tubules for fine tuning, and the loop of Henle is all about the overall volume of blood and therefore volume of urine, as well as the osmolarity of those things as well. And the other component, as we talked about, were the blood vessel components. And therefore, tubular components, therefore blood vessel components. They begin with the glomerulus, that highly specialized capillary that is part of the renal corpuscle. Then we have our efferent arteriole that feeds into our peritubular capillaries. However, as we also talked about, there are two main types of nephrons. There is a cortical nephron with small loops of Henle that are either entirely contained in the cortex or barely dip into the medulla. But there are, and they make up about 15% of the nephrons, these nephrons that have extremely extensive loops of Henle that penetrate deep down into the medulla of the medullary pyramids of the kidney, and these are called the juxtamedullary nephrons. And on them, we have these specialized lattice-like capillaries called the vasa recta. So again, our four blood vessel structures of the nephrons are the glomerulus, the efferent arteriole, the peritubular capillaries, and the vasa recta. What we left off in the last class talking about was then to help to auto-regulate the activity of this nephron, we have this juxtaglomerular apparatus. This juxtaglomerular apparatus is comprised primarily of three main types of cells. We have the macula densa cells, which are tightly coiled, tightly packed, columnar cells in the distal convoluted tubule that have chemoreceptors and osmoreceptors on them. We have the granular cells, which are the specialized smooth muscle cells of the afferent arteriole that are a combination of both smooth muscle and endocrine cells containing large granules containing hormones and hormone-like proteins, and the actin-filled mesangial cells, which are able to dynamically change their shape. So those three cells work together for that. The other thing that does remind me about is I believe I posted, and I wonder if I can switch to that. I don't know how I can do that now. Here, maybe if let's not worry about it. I believe, and I'll check during the break, that I posted some histology slides for you that showed that macula densa cell better than the pictures we looked at in the last class. So we have that for us as well. So that's the anatomy we spent a lot of time talking about. Our goal now is going to be to talk about the process of the formation of the urine, or again, really the processing of the blood. And that's really what we want to see. And notice here, we've got this more simplified illustration that kind of shows this process. And I actually want to turn it on its head to make a little bit more sense of it. There are three steps in the process of filtering the blood. And again, when we talk about formation of the urine, that's really what we're talking about. What we're really talking about is the processing and filtering of the blood, because that's really what we care about. We don't care so much about making urine. Making urine is just the waste product from this processing of the blood that we want to do. So let's switch back to our whiteboard so we can get a little bit more elbow room to do some drawing and writing. Go ahead and clear that. And what we can do is we can kind of do a similar picture to what we just saw before. I'll cheat. Oops. So here, and I need this to be red. Perfect. This right here, and we'll put a big fancy G inside of it, is our glomerulus. All right, we know it is fed into, and I'll do it in pink because I want to emphasize that it is not considered part of the nephron, but we know feeding into our glomerulus is going to be that afferent arteriole, and we'll just abbreviate that AA for right now. But again, that is not part of the nephron. We then have the tubular components, and we'll make those green. We have Bowman's, oops, no, that's not what I wanted to do. We have Bowman's capsule, and again, we're going to abbreviate all this stuff to make it simple. Surrounds the glomerulus, and again, remember these things collectively are known as the renal corpuscle. That then feeds into the tubular components. And those tubular components are comprised of the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. And again, we know they're all twisty and turby and things along those lines. And yes, remember Bowman's capsule and glomerular capsule are exactly the same thing. Glomerular capsule is a more descriptive term. It is the capsule that wraps around the glomerulus. You know, Bob Bowman was the first person who discovered it, so that's why we call this Bowman's capsule. That's also why we call this in here a Bowman's space. But glomerular space and glomerular capsule are perfectly acceptable terms for this as well, absolutely. Remember also that our, and again, I'll change the colors to emphasize it, our distal convoluted tubule is also going to then feed into our collecting duct. And remember that collecting duct is not a part of the nephron, just like the afferent arteriole is not. And then, of course, the end product at the end of this coming out here is our urine. So anything that is in our collecting duct and expressed from our collecting duct at the end of the process is the urine. Here in our afferent arteriole, we have the quote-unquote dirty blood. That is located here inside the afferent arteriole as it enters into the nephron. From there, it enters into the glomerulus, our first structure of the nephron from a blood vessel standpoint. That then feeds into our efferent arteriole. And that efferent arteriole is then going to feed into the capillaries that surround the tubules. And so, of course, those are the peritubular capillaries and the vasa recta. At the end of this process, in the blood vessel, after it's passed through the nephron, we then have our quote-unquote clean blood. So that's really the goal. That is our process. Our process is to start with dirty blood entering into the nephron, and then we pull out all the waste, all the extra, all the unwanted material, and release that from the body in the form of urine, where in that process we then clean the blood, make the blood nutrient appropriate. Well, not nutrient appropriate, that's what the liver does, but make it ionically, osmotically, pH appropriate. We then have that clean blood, and that clean blood is the end goal of the kidney. Now, as was mentioned, this whole process takes place in 3 steps. The first step is what is known as glomerular filtration. So this is step 1, so we should go ahead and go back here, make sure we emphasize that. Glomerular filtration, this is the first step in this process. And based on the name, not surprisingly, this occurs in the renal corpuscle only. All right, remember the renal corpuscle, and we can cheat and put a square around it, is basically this right here. Again, the renal corpuscle is the glomerulus, Bowman's capsule, and Bowman's space. So these things collectively are our renal corpuscle. Now, this glomerular filtration process, let's line this up here, is a passive process. Remind me again what that means?
No ATP is used.
Yes, that is correct. Oh, and I just saw your, Lauren, I'm sorry, I just saw your question. Yes, glomerulus and glomerular capillary is interchangeable terms, absolutely. So yeah, the glomerular just is the adjective to describe the capillary. Glomerulus is the noun that gives it its name. Yeah, this is a passive process. No ATP is used in this process, which is great, right? It doesn't use ATP, that's excellent. But the key, if it's a passive process and you're not using ATP, is that it cannot be selective. What I mean by that is we don't get to determine what comes out of the capillary. Really, the only specificity, the only determinant, let's go back, is size. Basically, what that means is all small things leave the capillary. And that is going to be both quote unquote good things and quote unquote bad things. Right? The example I give of this is that, again, spring break is coming, and back in ancient times, people used to go away on spring break, right? So you'd go to Mexico, or you'd go to Tahoe, or you'd go to New York, or wherever it is. Not allowed to leave and go anywhere now. Now your spring break gets to be you get to leave your bedroom and go to the living room. But in ancient times, you would go away for the week, and when you came back, you would have a big, huge, massive stack of mail. And typically when someone first gets their stack of mail, they sort it, and they sort it solely by size. All the small things go onto one side, all the big things go on the other. And that's because most of the small stuff is important stuff. Things like the bills, things like the birthday card from grandma with the check in it, and all those kind of things along those lines. And the big things are the flyers and the advertisements and the things that aren't important. However, it's not a perfect, just dividing it by size isn't really a perfect. There's plenty of small envelopes that are advertisements that people are sending you. And there's also plenty of big things that could be that, that large package from Amazon, or, that acceptance packet from your four-year university that you're going to, or any of those types of things like that. So we really can't be selective. In this case, basically, as we saw, there is that filtration membrane, and we'll take another look at that again today. We haven't done that yet, but I've got some great pictures that we'll take a look at that today. Anything that is small enough to fit through there is going to be filtered out. So that means both good and bad things, right? A good thing like sodium, a good thing like an amino acid, a good thing like a glucose. All of those are things that are good and we want, but they're small and they're going to sneak out, right? There's also plenty of bad things that are going to sneak out: ammonia, urea, all sorts of other metabolic waste and things along those lines as well. So again, the key is this is not selective. It is just determining basically by size. Anything that's small sneaks its way out. I need a little bit more room, I think, for the next one. All right. So again, this glomerular filtration is the first step in the process. It occurs only here in the renal corpuscle. It is a passive process, which means it cannot be selective. The only determinant is the size. And the other thing, and I guess we should emphasize this here, the direction. In this case, and again, we can think of it two ways. It is things are moving from the blood to the tubule. All right, so that is the way. So out of the blood into the, from the blood into the tubule, or I guess we should be more specific, from the capillary into the tubule. Or we could think of it the other way around as well. It is going from the blood to what we are calling the filtrate. Again, remember we talked about this last time. When we are first making this, and during the course of a day, we make about 200 liters of this filtrate. Filtrate is not urine. Filtrate is just the water and the small things that are pushed out of the glomerulus. So it is going from the blood to the filtrate, or from the capillary to the tubule would be the way to think of that. All right. We've now pulled out a big chunk of stuff, but it's a mixed bag. There's plenty of small good things that snuck out into that filtrate as well. And by the same token, there are plenty of big things that could be big waste, big metabolic waste, proteins, toxins, things that we want to get out as well. So we still need to continue our processing. And so while here in our renal corpuscle, our first step, the glomerular filtration, takes place, The second step in this process is tubular reabsorption. Tubular reabsorption occurs between the rest of the tubules and the peritubular capillaries. And of course, that also means the vasa recta as well. So that's step two. So while step one occurs here, nice black one there. And again, because we want to emphasize the direction, we can do an arrow. It is coming out. Step two, well, we'll do that in a second. Step two occurs in the tubular reabsorption, occurs between the rest of the tubules and the peritubular capillaries, which also includes the vasa recta. One of the big differences between glomerular filtration and tubular reabsorption, aside from the location of them, is that tubular reabsorption is a selective process. And to be selective, it needs to be active. It is going to be an active process. It is going to use ATP. And the advantage of using ATP is that it can be selective. Now, with a name like tubular reabsorption, obviously the direction we are going to be going with tubular reabsorption is going to be out of the tubule back into the blood. All right, so when we think in terms of direction, we are going to go from the tubule into the blood vessel. Well, really into the capillary, I guess, which should be consistent. Or again, from the filtrate to the blood. And if that is the direction we're moving, what type of materials do we want to bring back into the blood? All right, this is where we want to, all right, get the good stuff, things like ions. All right, we want to bring the quote unquote good stuff back into the blood. So that is going to be our tubular reabsorption. Right, coming out of the blood back, out of the tubule back into the blood. Remember, anything that stays in the tubule is going to become urine. So anything good we want, we need to be able to pull back out. Now, as we talked about, this allows us to get the small things that are good back into the blood, but we still have the issue of there being large things in the blood that didn't come out, or maybe some small waste that snuck back in. So we need a third process, and that third process is going to be tubular secretion. And this is step 3 in the process. Like tubular reabsorption, this occurs between the rest of the tubules and the, oh, it's supposed to be peri. That's why I did wrong. Peritubular caps, including the vasa recta. Now, one of the things that I want to emphasize is we are putting these in order, giving them numbered order, 1, 2, and 3. 1 definitely occurs first. So when we talk about these processes, 1 definitely, glomerular filtration has to occur first. That has to occur first. However, 2 and 3, oops, I didn't want that to be green. Let me black. Oh well. glomerular filtration rate has to occur first, but tubular reabsorption and tubular secretion occur at the same time. And the other important thing to remember about 2 and 3, remember, most of the processing, both the reabsorption and the secretion, occur in the proximal convoluted tubule. So that is true for both tubular reabsorption and tubular secretion. The majority of these things occur here in the proximal convoluted tubule, both the tubular reabsorption and the tubular secretion. So the majority of both of those are occurring here in the proximal convoluted tubule. Let's move it over here to emphasize that. Perfect. In many ways, tubular secretion is identical to tubular reabsorption. The only difference is the direction. They actually use very similar mechanisms. They are going to primarily be active processes. So again, it is going to use ATP. We're going to use the same type of pumps, the same types of transporters that we saw before. So that's going to be the same. However, the reason for it, because it's going in the opposite direction, is we want to basically remove, in this case, the bad stuff from the blood. And so, of course, the direction this is going to go is from the capillary into the tubule, or from the blood into the filtrate. All right. So this is what's going to be happening. We have this movement, and let me do that and that. 3 processes: glomerular filtration, tubular reabsorption, tubular secretion. Glomerular filtration is a passive process that occurs in the renal corpuscle, where, again, it's non-selective, so basically just everything small and water comes out of the capillary into the tubule, and this is where we produce our filtrate. Then we process that filtrate. Because remember, we start with 200 liters of filtrate during the course of the day, but by the end of the day, we produce somewhere between 1 and 2 liters of urine. So there's a massive amount of processing that takes place of that, and that occurs out here in the rest of the nephron with our tubular reabsorption and tubular secretion. And again, the majority of that occurs here in the proximal convoluted tubule, with less of it occurring in the loop of Henle and less of it occurring in our distal convoluted tubule. And again, even though we are listing them sequentially, even though we're going to talk about them sequentially, it is important to remember that tubular reabsorption and tubular secretion are occurring at the same time. All right, let me take a picture of the screen here. So hopefully that can help us to understand this, and I'll be posting these on Canvas. I've been doing that with my 430 class, and I think it's been useful. So I'll do that here as well. We'll post that onto our Canvas site, so you guys will have that to look at. All right, questions on this? All right, then let's go back to our lecture and go through the same thing again here. Again, we have our glomerular filtration rate occurring just here in the renal corpuscle, where we have the solution coming out. And again, it's that passive process. Tubular reabsorption, where things are moving from the tubule back into the blood. Reclaim all that good stuff that snuck its way out. And tubular secretion, where's my mouse? There it is. Where things are being released from the blood into the tubule so that it can be expelled from the body. Because at the end of this process, where's my annotation? there it is. We are going to have processed blood, and we are going to have urine. All righty. Now, let's learn everything you ever wanted to know about glomerular filtration and more. Again, glomerular filtration, as we talked about, is occurring in the renal corpuscle. And again, our goal here is to produce filtrate. $200 liters of it. And it is very, very different from urine. Filtrate is essentially our blood plasma, you know, the water that comes out, but there's no formed elements, there's no large proteins, and like I said, it is a collection of both good and bad. The only determining factor as to whether you get into the filtrate or not is essentially the size. If you're small enough to sneak through the cracks, then you come out, whether you're a good thing or a bad thing. Like I said, we don't get to be selective. It's just sorting by size. That's really what this process is. We are basically just sorting by size. I like that. I'm gonna write that down. All right, that's all we're doing. We're really just sorting material by size. Then once it's sorted by size, it's going to be easier to deal with. Once we sort it by size, then we have to process it, and we go from 200 liters of filtrate to, as we talked, oops, go back, to, as we talked about, we go to 1 to 2 liters of urine. So that's a massive amount of processing that takes place, and that urine is basically metabolic wastes, excess unneeded substances, toxins, which I guess still fall into unneeded substances, and a little bit of water. All right, so again, there's a very big difference between filtrate and urine. And remember, it does not become urine until it leaves the medullary pyramid, not leaves the nephron. Because remember, when it leaves the nephron, excuse me, that filtrate enters into the collecting duct. And remember, as we talked about, the collecting duct has those principal cells, has those intercalated cells that are still capable of processing it. There's still going to be some fine tuning that takes place there. So it isn't until it leaves the collecting duct, is expressed out the papillae into the minor calyx. And once it reaches the minor calyx, then it is urine. But from the time it is expressed in the glomerulus till that, it is filtrate. Because if we can process, it's filtrate. All righty. Like I said, let's talk about this glomerular, clear my drawings. Filtration, again, it is a passive process, and it is going to rely on the blood pressure of the glomerulus to produce the filtrate. About 10% of the blood plasma is expressed out of our glomerulus. And like I said, we make about 200 liters of filtrate during the process of the day. And then, excuse me, end up with about 1 to 2 liters of urine. As we also talked about being a passive process, there are things we want to do to maximize the efficiency of this. One of those things is to have an incredibly thin filtration membrane. We've already identified it, and this picture does a nice job of showing it, although we'll talk about it again and look at the pretty picture in just a second. Notice here we are seeing the fenestrations of that endothelium of the glomerulus. The glomerulus is a specialized capillary, and one of the things that's special about it is that it has that fenestrated endothelium to it. Simple, excuse me, simple squamous epithelial cells with fenestrations in them. And then we have the podocyte, the visceral layer of Bowman's capsules, that special podocyte cells with those foot-like processes that intertwine with each other. So we just have these tiny filtration slits that you can find in between. So here are the little fenestrations, the little slits between the finger-like extensions of those podocytes is the space where it sneaks out through. So it's a very, very small space. Huge surface area. Like I said, every kidney has over 1 million nephrons in it. So we have a massive surface area to these things. And remember, one of the other things we said that's special about the glomerulus is that it is a high pressure blood vessel. And so that high pressure blood vessel, that high pressure is going to affect the rate at which it expresses the filtrate. And we did the calculations for capillary exchange before, and we're going to do it again in just a minute. As I mentioned, this occurs at the filtration membrane. We talked about what this was last time. It is that fenestrated endothelium of the capillary. It is that visceral layer of Bowman's capsule, those podocyte cells, and their fused basal lamina. And in this case, the fused basal lamina of these two cell layers is known as the dense layer. Here we have the pretty illustration from your textbook that again shows our capillary, the endothelium, with those fenestrations in them. And here we have the foot-like processes of the podocytes with those tiny little filtration slits in between. And in gray, we have our fused basal lamina or basement membranes. But like I said, one of the things I love about your textbook is your textbook has these great electron microscopy views. Right? When you think about what it takes to make an electron microscopy view, you have to take a piece of tissue, you have to freeze it, you have to slice it really thin, you have to put it on a slide, mount it on that slide, and then put it in this special microscope that bombards it with electrons. It destroys the tissue when you do this, but as a result, we get to see these microscopic views of the proteins and the organelles and the structures inside of a cell that no light microscope would ever allow us to see. And that's what we have here. Here we have an electron microscopy view of the endothelium of our glomerulus. Here we see some of those fenestrations in it. Here we see those irregular foot-like processes of the podocytes, and then we can see the gaps in between them, which as we mentioned are the filtration slits. And here is the basement membranes of the two fused together. And you can see now why it's called the dense layer. Because again, those two basement membranes together form actually kind of the darkest and most continuous structure. And as was mentioned, I said it on the previous picture, I'll go back for it for just a second. The space in between is only about 6 to 9 nanometers in width. So only something that is less than 10 nanometers is going to be able to wiggle out of wiggle out of fenestration, wiggle through that filtration slit, and end up here in Bowman's capsule, or really Bowman's space would be more accurate, inside of Bowman's capsule, where it is then going to be processed. All righty. Questions on that? All right. Now, as we talked about our capillaries way back in the day, and let's go ahead and cheat and do this again. We'll make it red. We talked about pressures. Capillary exchange is all about pressures. And if you remember, there were 4 pressures we talked about in the capillary, but we only have to worry about 3 of them here in our renal corpuscle. So let's start first with our glomerulus. And of course, as we know, our glomerulus, because it contains blood, has 2 pressures to it. So let's do that. I'll move this a little bit that way. That will allow me to put this here more. Give me some writing room. Excellent. In our blood, in our capillary, if you remember, there were 2 pressures. There was the hydrostatic. I know as I write this, everybody groans because, oh my God, they hated that capillary exchange. Did poorly on the essay question of it and was hoping to never have to deal with again. But unfortunately, we do. That hydrostatic pressure of the capillary. So we have that to deal with again. And let's do that. So that is one pressure we have to deal with. But remember also, we have to deal with the osmotic. pressure of the blood. So there are two pressures that we need to deal with inside of that glomerulus: the hydrostatic pressure of the capillary and the osmotic pressure. Let's give myself a little bit more playroom for that. One of the things, if you remember, we talked about is that the hydrostatic pressure of the capillary is very high. It has a high blood pressure. If you remember, in a typical capillary, we know that at the arterial end, that pressure is going to be around 35, and at the venous end, it is going to be closer to 17. But remember, in our glomerulus, we talked about that pressure being 55 millimeters of mercury. It is a very, very high pressure. And of course, when we think of these pressures, we have to think of the directions that pressure is going to be pushing things. And in this case, that pressure is, remember, going to be an outward pressure. Oops, I don't want that color. This one. There we go. It is an outward pressure. Out of the blood vessel, out of the capillary, into the space. All right. Our osmotic pressure, remember, is an inward pressure. So let's pick a different color for that one. Oops, no. Undo that. No. I hate this stupid thing. I want to be back in the classroom. Delete that. Do that. Do that. This is where my OCD becomes a problem. I don't have a lot of it, but there's some things that it bugs me. All right, excellent. So again, that is an outward pressure. We have that there. Perfect. Now we want to change to green. Our osmotic pressure, remember, is an inward pressure, where again, it is the stuff that is in the blood that is going to draw water inward towards it here. And so all we need to do now is add a value to this. And it turns out the osmotic pressure of the glomerulus is about 30 millimeters of mercury. So again, when we did that capillary exchange, none of this is new information. We know that blood has a hydrostatic pressure, the force of the water pushing against the walls, and it is an outward force. The blood has stuff in it. That stuff draws water to it, so that is an inward force. And so then all we have to do is know the specific values of those in our glomerulus. Now, that glomerulus is then going to be expressing our filtrate. Oh, no, I don't want to do that. Let's go with black. Nope. Let's go with brown. I like brown. Into our Bowman's capsule. So here we have our Bowman's capsule. And again, glomerular capsule is perfectly acceptable. For whatever reason, I like saying Bowman. Bowman's is more fun to say. It's easier to spell. So either of those are fine. And remember, when we did our capillary exchange, we had the interstitial fluid. In this case, this wouldn't be interstitial fluid, whereas we have blood here inside of Bowman's capsule, we have the filtrate. But the filtrate is still water. The filtrate is still water with stuff in it. But here's the trick. The filtrate, right, the reason that those pressures of the interstitial fluid matter is because the interstitial fluid surrounds the capillary, and so it interacts with it. Once this stuff is expressed out of the glomerulus, the filtrate doesn't stay. The filtrate leaves and goes down the proximal convoluted tubule. And because it goes down the proximal convoluted tubule, there's no water here with any stuff in here to have an effect. So essentially, the osmotic pressure here in the Bowman's capsule is 0. And if it's 0, when we do our calculation, do we need to bother worrying about it, or do we even care about it? No, absolutely not. However, as we saw, and again, my drawing doesn't do a nice job of showing this, we know Bowman's capsule is tightly wrapped around that glomerulus. So think of it this way. If you're standing out in your backyard right now, watering your lawn with a hose, and you take your hand and put it most of the way over the top of the opening of that hose, is that going to change the pressure of the water as it's coming out of it? Absolutely, right? That's what you do. You're standing there with your hose, just watering the lawn, and your significant other walks outside. So what do? You take your thumb, you cover 2/3 of the opening, that increases the pressure, and you're able to spray your loved ones with the hose as they step outside, and then you go back to what you were doing before. So while there isn't an osmotic pressure, the close proximity of that barrier, of that Bowman's capsule, does produce a hydrostatic pressure. So we do have a hydrostatic pressure of the capsule. And that hydrostatic pressure of the capsule is going to be, need to move that. Cheat and put this down here a little bit more. The hydrostatic pressure of the capsule is going to be about 15 millimeters of mercury. And it is going to be an inward force, in which case it is going to be trying to push water back into our capillary. So when we are calculating, and that's really what this is going to be about again, our net filtration pressure, basically what our net filtration pressure is, and I'm going to abbreviate it up here, net filtration pressure, is going to be equal to is the outward forces minus the inward forces. That is how we calculate the net filtration pressure. It's how we calculated our capillary exchange as well. And in this particular case, what that means is our net filtration pressure is going to be equal to the outward forces. And how many outward forces are there?
2.
Well, in this case, how many are there? Just one, absolutely. It's going to be the hydrostatic pressure of the capillary minus the inward forces, and there are 2, the osmotic pressure of the capillary plus the hydrostatic pressure of the Bowman's capsule. And if we do the math on that, it is going to be 55 minus 35 plus 30 plus 15, which of course equals 55 minus 45, which of course equals 10 millimeters of mercury. And of course it is a positive 10, which of course, as we know, means it is an outward force. So our net filtration pressure is going to be a positive 10 millimeters of mercury, and it is going to be an outward force. So what that means is that that's a horrible stamp. All right, never mind. Forget that. The net effect of that, let's do it this way instead. It's going to, the net effect is going to be an outward force. We have that outward force of that as we go out. All right, questions on that? This net filtration pressure is going to determine the rate at which we make our filtrate. So let me go ahead and save this pretty picture again. There you go, screen has been saved, so excellent, that should help. And again, I can share those things. Excellent, and now we can clear the screen and we can go through it all again with the pretty pictures from your textbook. So again, our net filtration pressure is comprised of our glomerular hydrostatic pressure, which is 55 millimeters of mercury, and it's an outward force. Again, much, much higher than we saw in the normal capillaries, which are between 35 and 17. Our capillary hydrostatic pressure, again, the capsule, the Bowman's capsule, and that's 15, and that's an inward force. And our blood's colloid osmotic pressure, which, as we said, was 30. So when you put those together, our outward forces, and again, that's the key to this, it is outward forces minus inward forces. Our glomerular hydrostatic pressure minus the osmotic pressure of the blood and the hydrostatic pressure of the capillary. We put those together and we get a positive 10 millimeter change. So the net effect is an outward force, and so we get this outward pressure. And so plasma and anything small enough comes out and forms our filtrate. All righty? And so it is this outward force that is going to allow us to produce 200 liters of filtrate during the course of a day. Questions on that? All right, excellent. This is a good stopping point for our first break. Let's go ahead and take our first break. We will take a 10-minute break. That means we will return at, what will that be, 9:10. And again, I'll remind myself to start the recording. So again, any questions? All right, then I will go ahead and pause the recording and we will take a 10-minute break. See you in 10 minutes. All righty, let's go ahead and dive back in. So, as I mentioned, at the last, before the break, we calculated our net filtration pressure. And now that we have calculated our net filtration pressure, put that that way, we can understand that that is an outward force, and that is what is producing our filtrate. Our net filtration pressure is what determines our glomerular filtration rate. Basically, the glomerular filtration rate is the volume of filtrate that is produced during the course of 1 minute in time. For males, that's about 125 milliliters per minute. For females, that's 105 milliliters per minute. And again, of course, the primary reason for the difference is, of course, as always, men are better. Okay, maybe not better, but at least typically bigger. And so again, slightly different. But again, you take 125 milliliters per minute times 60 minutes times 24 hours a day, and you get pretty much that 200 liters of filtrate that we produce during the course of a day. Now, again, we talked a little bit about this in the last class. The rate at which we make filtrate is vitally important because, again, basically, we are taking this filtrate and putting it on that conveyor belt so that when it's on that conveyor belt, it can be processed. And the key to remember about this is if we produce too much filtrate, or you can think of it in terms of we produce filtrate too fast, then as a result of that, what's going to end up happening is we are not going to have as much time to process it. And so what is going to occur, the effect of that is that our filtrate will be under processed. We are not going to be able to process it nearly as efficiently, and our filtrate is going to be under processed. Conversely, if it's too low, then what happens is we're going to produce too little filtrate, or again, we are producing it too slowly. And that can be a problem as well, because if it's too slow, what's going to end up happening is we're going to have too much time to be too critical, and our filtrate will be over-processed. And both of these are concerns. We don't want to over-process, we don't want to under-process our filtrate. We need to make sure we keep a consistent rate of filtrate production so that we are doing the right amount of processing of our filtrate. This then, of course, is a vitally important process. And luckily, there are 3 ways we can regulate it. Now, again, as we talk about how we determine the glomerular filtration rate are going to be responsible of 2 factors: blood flow and the surface area, right? If blood flow increases, well, let's even be more specific. If the amount of blood in the glomerulus increases, right? If we have an increased blood flow, then that is going to mean an increase in filtration that is going to take place. And it's the same thing for surface area as well. If we have an increase in surface area, then we are going to get, then that's going to, let's do it this way. Infiltration. And of course, the opposite is true as well. If less blood goes to the glomerulus, less filtration is going to occur. If less surface area in the glomerulus, then we are going to have less filtration occurring. We've kind of talked about some of these methods, but now's our chance to get more in depth. So let's go ahead and switch the screen and then clear our drawings. And we can talk about that. Now, obviously, these mechanisms are going to be both intrinsic and extrinsic. Intrinsic, of course, as we talked about, means that it is contained within the kidney, and really in this case, as we've talked about, inside of the nephron. So some of it are going to be local, right? And that's another way to think of this. Local effects, whereas extrinsic primarily means from outside of the kidney. And if we're talking about outside of the kidney control, there are, of course, really 2 primary ways we do that: hormones and our nervous system. However, as we talked about in the last lecture, what branch of the nervous system is primarily involved in controlling the activity of the kidney?
Sympathetic.
There you go, absolutely, because it's not intuitive. But it is actually the sympathetic branch of the autonomic nervous system that is going to be part of that extrinsic outside control. So we have these 2 things. We've kind of already talked a little bit about the intrinsic control, and now we'll get a chance to talk about the others as well, but we'll go over it again as well. So let's move these things up and out of the way a little bit so a little bit more pretty. Excellent. And then we can talk about these. Let's talk about the intrinsic first. This is our renal autoregulation, and in fact, there are actually 2 types. So let's go back. Do I have room to sneak that in over here? Excellent. There are 2 types of intrinsic control mechanisms. The first of these is what is known as the myogenic mechanism. Pressure changes in the afferent arteriole cause changes to our glomerular filtration rate. Let's do a little bit of drawing for this. Where do I want to do this? Actually, let's go ahead and go to the whiteboard to do this. Go ahead and clear all of that. All right, for our myogenic mechanism. Basically, again, the key concept here are pressure changes in the afferent arteriole. This is what we are monitoring, and those are going to change the filtration rate. All right, let's do some quick drawing for this. We have our afferent arteriole. And what do I want to do? Is actually I want to move this down so it's out of the way. We can have the picture down here, and then we can talk about it above. So this is our afferent arteriole. It feeds into our glomerulus. Excellent. So let's put some labels on these. Our afferent arteriole. Our efferent arteriole. And then this, of course, is our glomerulus. Or again, as someone mentioned, the glomerular capillary. Oops. Excellent. All righty. Now, if you think about it, as we've talked about, you have 1 million nephrons in each kidney, and blood is flowing to them, but the blood pressure to every single one of them isn't going to be identical, right? Some may be slightly higher pressure, some might be slightly lower pressure, and that's even while you're just sitting there calmly. Every time you change body position, you stand up, you lay down, you stretch, you go for a walk, there are dynamic changes that are taking place in your blood vessel. And so what happens is the pressure of the blood coming into our glomerulus, and of course it's coming in via the afferent arteriole, is constantly changing. Right? Again, our bodies are dynamic, the system is dynamic. That should not be surprising to anybody that that is going to take place. So what that means is we need to be able to constantly adjust the diameter of the afferent arteriole to be able to maintain a constant flow. Things in my way. There we go. Because that constant flow determines a consistent glomerular filtration rate. Let's see some examples of how this might work. Let's say for starters that because you stood up or you started walking or something like that, there is a drop in blood pressure in the afferent arteriole. All right, let's give myself room to write this. So what happens is the blood pressure, let's change colors now. The blood pressure here inside of our afferent arteriole drops. First of all, the big question is, how would we know that? Well, blood pressure, remember, is the force that is pushing against the wall of the capillary. And so if the blood pressure drops, that basically means that there's less force on the wall, and there's going to be less stretch. All right? So again, there's less force of the blood pushing against the wall, and so there is less stretch of our afferent arteriole as a result of that. Luckily, we have baroreceptors, which are basically stretch receptors. In the afferent arteriole. And with those stretch receptors in the afferent arteriole, they are going to be stretched less. And as a result of that, they will produce fewer action potentials. Now, this is an intrinsic system, remember. So that means it's a local effect. What happens in our afferent arteriole, when there is less stretch, that means there's less blood pressure coming to the glomerulus, and we want to maintain an appropriate glomerular filtration rate. So if there's less stretch because there's less blood pressure because of the afferent arteriole, the pressure has gone down, we need something to be able to increase the amount of blood that's getting in there. So our afferent arteriole, when it stretches less, the goal of it is to get more blood into the glomerulus. If the blood has less pressure to it, we need a larger volume of blood in the glomerulus so we can maintain a consistent filtration rate. And so what would our afferent arteriole need to do to get more blood into the glomerulus?
Expand in diameter.
Absolutely, absolutely. What it is going to do is our afferent arteriole dilates. If it dilates, then what that causes is more blood to flow to the glomerulus. Right? We get an increase in blood flow, and that increase in blood flow is going to counteract the drop in pressure, and that way we will be able to maintain an appropriate glomerular filtration rate. So when there is less stretch, our afferent arteriole responds by dilates. Dilates, and when it dilates, that increases the flow. So now the pressure coming into the glomerulus may be lower, but we have an increased flow, right? We have more, because again, remember, what matters is that blood pressure inside of the glomerulus. We keep using the example of you guys bouncing against the walls, and we count the number of times that occurs, and that's the blood pressure. If I cool off the room and you guys get all slower in your movement, you are going to be bouncing against the wall fewer times, and so blood pressure would drop. But if I bring 10 more kids in there, and all of you are moving around more slowly, but because there's more of you, there's still going to be the same amount of bouncing at the wall. So our blood pressure goes down, we increase the blood flow, and the net effect is we're able to maintain our appropriate glomerular filtration rate. So we're able to counteract that drop in blood pressure by increasing the flow to the glomerulus. and maintain that consistency. And again, this is a dynamic change. All of this effect is happening right here locally in the afferent arteriole. There's no outside stimulus, and again, that allows each nephron to adjust its own afferent arteriole as needed to maintain this glomerular filtration rate. Does that make any semblance of sense?
Yep.
Excellent. All right, if that makes sense, then we can do the opposite as well. If there is an increase in blood pressure, in the afferent arteriole, then we want the opposite to occur, right? If you think about what would happen in this case, with an increase in blood pressure, our stretch receptors would be, or let's say it this way, our blood vessel, the afferent arteriole, would stretch more. Our stretch receptors would produce more action potentials. And as a result of that, in our afferent arteriole, because our goal is to now, we want the blood pressure is higher. With a higher blood pressure, our goal is going to be to get less blood into the glomerulus. Since the blood is at a higher pressure, we need to make sure that there is less of it getting into the glomerulus. And so, of course, the result of that is our afferent arteriole will constrict. That means we are going to get less blood flow going into the glomerulus. But remember, it is at a higher pressure, right? It is still higher pressure blood. So if you guys are instead running around the room and bouncing into the walls, then I need to pull 10 of you out of the room so that that pressure stays consistent. And again, we'll be able to maintain the appropriate glomerular filtration rate. So again, we are going to get dynamic changes in the afferent arteriole to be able to correspond to those changes in pressure so that we can get the right amount of blood, because it's all about flow, right? Those are the two things that we keep talking about, flow and pressure. Those are going to be the things that matter. So if our pressure changes, we have to change the flow to compensate, right? And again, this is going to be an important fact because we're going to see that this can be a little confusing. The key is we need to correlate flow and pressure. All right, that is going to be the key to this process. The key, is that we want to correlate our flow and pressure. In this case, with this one down here, what's happening is that here we are getting pressure changes. So because pressure is changing, we have to change the flow. I make a point of emphasizing this because when we get to the intrinsic controls, the extrinsic controls, one of the things that we're going to see is that there's going to be a change in flow to our glomerulus. And if we change the flow to the glomerulus, guess what we have to change? That's right, the pressure. All righty. So this is the pretty picture for our glomerular filtration. Myogenic mechanism, that affects our glomerular filtration rate, our first intrinsic process. So let me go ahead and save that pretty picture. And then we can go back to the lecture. I need to bring that back up, and I need to bring that back up. Excellent. All right, so again, pressure changes in the afferent arteriole. So I need to bring that back up. The second is our tubular glomerular feedback. This is the stuff that we talked about at the last class. Our tubular glomerular feedback is what is occurring in our juxtaglomerular apparatus. Remember, this one is where we were talking, and I'll just write it here because I think it's we did a good job of describing this in the last class. But again, the take-home message is we have those macula densa cells that are in the distal convoluted tubule. And again, they are doing that quality control of our almost urine. Right, so they are checking the condition of our filtrate to see if it is being over or under processed. Remember, if it is being over processed, that means we are not making enough filtrate. And if that occurs, our macula densa cells release more nitric oxide. And that nitric oxide has two effects. The first thing it does is dilates the afferent arteriole, which is going to increase the flow of blood to the glomerulus. The second thing it is going to do is that nitric oxide stimulates the mesangial cells to build more actin. And if they build more actin, the cells get bigger. And that pushes the glomerulus out. And as it pushes the glomerulus out, that increases our surface area. And if we increase the surface area, or if we increase the flow, the net effect of all of these things is that we are going to get more filtrate. All right, because that's our goal. We've been under processing it. We need to make more. And so that's what happens there. Conversely, if our macula densa cells see that it is under processed, that means we are making too much. Right? And so they release less nitric oxide, which causes two effects. Constrict the afferent arteriole, and that decreases the flow into the glomerulus. And our mesangial cells break down actin. Cells shrink in size. That collapses glomerulus, and the surface area decreases. And again, the net effect of that is we make less filtrate. All right, the net effect of this one above is we make more filtrate. Again, I think we talked about that in pretty good depth last time, but this is a good reminder of those effects here. So that is basically those three regulatory cells, the mesangial cells, the macula densa cells, and the granular cells. And of course, it's the granular cells that are constricting the afferent arteriole or dilating the afferent arteriole. So I guess we should remind ourselves of that. So, okay, so I did that. And so, of course, again, it is the granular cells or juxtaglomerular cells that are dilating or constricting. And one of the important things to remember, and this will get to the next thing, when these granule cells dilate and constrict, that also is going to affect their hormone release. Because remember, these granular cells are both smooth muscle cells and endocrine cells. So we're going to see that change in that as well. And that's going to lead to part of our extrinsic mechanisms. Again, I know I went through that kind of quick, but I think we had talked about that in pretty good detail in the last class. If it's still confusing for anybody, I'm happy to go through it a second time slower to make sure this makes sense. But again, I don't want to beat the dead horse if we talked about it well last time. So any questions on what's happening with the juxtaglomerular apparatus? All right, a blinking screen tells me that you guys have all mastered this material. So let's move on from this. So again, we'll clear our drawings for this. And again, maculodensa cells, quality control, releasing that nitric oxide. That affects our mesangial and our granular cells. All right, what we need to talk about next is our extrinsic mechanisms. And as we hinted at, there are two, neural and hormonal. First, we will talk about the autonomic. Again, as we already mentioned, it is the autonomic that is controlled by the sympathetic nervous system. One of the big differences between what we see here with our sympathetic nervous system and with that myogenic mechanism is that our sympathetic nervous system is going to affect both the afferent and the efferent arterioles. So what we're going to see is changes in both. Let's see what I have here. And then, of course, it's going to stimulate the granular cells to release our hormones. So let's talk about this autonomic regulation. For this, we need a little bit more elbow room to be able to do that. Oops, no, not what I wanted. That's what I wanted. So what we're going to do here is go back to our whiteboard. What I'm going to do is I'm going to cheat and leave my picture so I don't have to draw that again. There we go. So we have our pretty picture, just like last time. And again, what we want to talk about is, again, our goal is to maintain an appropriate or consistent, I like that even better, glomerular filtration rate. And again, the two things we have to are the flow and the pressure of the blood. All right. Now remember, with our myogenic mechanism, it was the pressure of the blood that was changing. And so we needed to compensate by affecting the flow. Here in our sympathetic mechanism, our issue here is that we need to respond to changes in blood flow to the kidneys. And if we're going to respond to changes in blood flow, what do we need to modify to respond to that? We We need to change the blood pressure. All right, that is really the key to this process. It's kind of the opposite of what we saw in the myogenic mechanism, because what we're accounting for is changing. In this case, what's going to happen is we are going to have changes in the blood flow to the kidney. And so what we need to be able to do is we need to change the blood pressure in response to that. And if we can do that, we can maintain a consistent glomerular filtration rate. Now, let's think of this in terms of three conditions. Normal resting conditions. As we talked about in normal resting conditions, while you're sitting here calmly in the classroom, or in your bedroom, or wherever it is that you're doing this, outside by your pool, whatever tickles your fancy. Under normal resting conditions, we have a large blood flow to the kidneys. Right? As we talked about, while you're sitting here calmly listening to my lecture, 25% of your blood is going to your kidneys every minute to be processed. So we have a large blood flow to the kidneys, and with plenty of blood flow, we can maintain a normal glomerular filtration rate. So there is no need for adjustment. Again, one of the things we talked about is that there isn't a dramatic, dramatic, I like the word dramatic. There isn't a large parasympathetic input to our kidneys. Yes, filtering the blood seems very much like a housekeeping type of process, but that housekeeping type of process doesn't need any stimulus from this parasympathetic nervous system to tell it to do it. It's going to normally do its job, and that's not a problem. What becomes a problem is when we take our 10-minute break. When we take our 10-minute break, you get up and you run downstairs to your kitchen so that you can make yourself a cup of coffee, or you run to the bathroom, or you stand up and do 10 jumping jacks to get the blood circulating back to the flow of your legs again, or things along those lines. So while you're sitting here, there's plenty of blood flowing to the kidney. The problem is when we get a normal level of activity. Or let's say it this way, I like it better this way. Moderate level of activity. With a moderate level of activity, our body redirects blood to other areas, right? Like the liver to get glucose, like the muscles, right? We constrict it in the skin. Things along those lines so that we can maintain that moderate level of activity, which is all a great, awesome thing. But the net effect of that is now less blood, or let's be more specific to make sure, again, stay with the terms we've been talking about it, less flow, right? And again, if you think about it of overall volume, sorry, I'm having to move my things around. That works. Less flow or less volume of blood goes to the kidneys. And that's an issue, because if the flow decreases, then our glomerular filtration rate is going to decrease as well. And we don't want that. Every time we stand up and walk around, we don't want our kidneys to be less efficient. So when our moderate amount of activity, and of course, if we have a moderate amount of activity, we have an increase in our sympathetic activation. And what that sympathetic activation does in the case of our kidneys is it causes a constriction both the afferent and the efferent arteriole. me go ahead and abbreviate that A of the afferent to make sure we understand that. So it's going to constrict both the afferent and the efferent arteriole. Let's think about what kind of an effect that would have. Let's go ahead and come down here, and now we'll change colors of our text. Right? Again, the stimulus is that there is less flow in the afferent arteriole. So our sympathetic nervous system stimulates the afferent arteriole to constrict. Notice there's already less blood coming in here. We have a less volume of blood. So what we need to do is increase the pressure of that blood. And so if we're going to increase the pressure of the blood, constricting that afferent arteriole is going to increase the pressure. Because as we know, if you decrease the volume, that is going to increase the pressure. And I cheated to give myself a little space there so that I can put in my arrows. So if the volume goes down, the pressure goes up, right? Now, I can already hear what you're saying. Wait. When we constricted the afferent arteriole before, that decreased the blood flow, right? But remember, there's already less blood in here. So because there's less blood in here, there's less blood in here, and this less blood has a lower pressure to it. So when we're constricting the afferent arteriole, we're not affecting the volume of blood in this case because the volume of blood is already changed. So what we are affecting is the pressure of that blood. So again, as a result of that, we get less blood in the glomerulus, but that blood at least has a higher pressure. And so there's less blood in here, but it has a higher pressure, and that's going to help to maintain a glomerular filtration rate. But it can't do it on its own. And luckily, remember, our sympathetic nervous system is also going to constrict our efferent arteriole. So again, our sympathetic nervous system stimulates the efferent arteriole to constrict as well. In this case, when we constrict it, it becomes more narrow. And if it becomes more narrow, this is going to decrease the flow of blood that is able to leave the glomerulus, right? I love to use the example of Davis. When you are driving down 80, coming from San Francisco, right? It's 3 o'clock in the morning on a Wednesday. There are no other cars there, so you're able to go 115 miles an hour. Then you hit Davis. And even 3 o'clock on a Wednesday during quarantine, right? There's a ton of cars there and the traffic slows down to 15 miles an hour. There's that massive congestion, right? And the congestion is because you go from the 15 lanes that are Interstate 80 down to 2. And that's what we're doing here. By constricting the afferent arteriole, we decrease the flow, and basically what we produce as a result of it is congestion in the glomerulus, right? Or you could even think of it as turbulence in the glomerulus, because now it is harder for it to get out. This congestion slows the blood in the glomerulus, which means there's going to be more blood in the glomerulus. And of course, if there's more blood, we get more pressure. So notice the effect of constricting both of these is that we have less blood overall coming to the glomerulus because we are now being active. But by constricting the afferent arteriole, we increase the pressure of the blood. By constricting the efferent arteriole, we maintain more blood volume in the glomerulus, which gives us more pressure. And so we can maintain a consistent glomerular filtration rate even with less blood. And that's really the huge advantage of this process. The huge advantage of this process is with less volume of blood going to the kidneys, we can still maintain our normal rate of filtration. And that's why it's just the sympathetic, because it is when we're active that we need to be able to modify and adjust what we're doing to make sure that we can still filter the blood efficiently, even when there's much less blood going to the kidneys. And that is great, and that is awesome, except for one small issue. This works great when there is a moderate level of activity, but despite, and again, I will say that not a lot of you have taken that practice lab exam yet, although I strongly encourage you all to do it. But so far, I don't think anybody has gotten the question right. And again, don't worry, you still get the points even if you don't get the question right. What, of course, is the scariest, most scariest possible thing in the entire world?
Bear with an axe.
There you go, absolutely. It is a bear with an axe. So if that bear with an axe comes at you, again, you have to outrun somebody in your household, or you need to fight that bear with an axe. And in those type of extreme situations, and let's change the color of this to emphasize this. With extreme levels of activity, like running from that bear with an axe, we are going to get a dramatically decreased level amount, let's say it that way, decreased amount of blood flowing to the kidneys, right? So with an extreme level of activity, what happens is we get a dramatically decreased amount of blood to the kidneys. Now, obviously, with an extreme level of activity, we are going to get a high level of sympathetic activation. But do you think we're going to be able to squeeze that afferent arteriole and that efferent arteriole enough to compensate for a dramatic decrease in blood flow to the kidney?
Nope.
No, so it's not going to be enough. At that high level, even though we have a high level of sympathetic activation, at extreme levels of activity, it's not enough. And so the result of that is during those extreme levels of activity, there is a decrease in the efficiency of our kidneys. Filtration levels do drop. Now, the good news is, in most cases, that type of extreme stress only lasts a short time, right? You can only sprint away from that bear for, 5-10 minutes. You can only fight that bear for 5 or 10 minutes, right? So again, typically that kind of extreme stress that decreases the efficiency of our kidneys only lasts for a short period of time. Except when now suddenly all your online classes or all your classes are now online, and everybody's worried about every single sniffle or cough or tickle in their throat because of the coronavirus and all these other types of extreme stress. The problem is when we deal with chronic stress, that chronic stress has the potential to be able to have long-lasting impacts on the efficiency of our kidneys. When we're dealing with those constant stressors that are constantly doing it, that sympathetic activation can affect, because again, we still have those same biological responses where blood goes to our liver and to our adipose and to our muscles when we're stressed, because our body's saying, stress, stress, deal with it from running or fighting. But those aren't the things that are causing the stress anymore, and that's not how we respond to them, right? We can't, go outside, go to the gym and exercise. We can't run from the coronavirus, right? Now they're talking about the 6 feet's not enough. Some of that, the research on that is very antiquated, and the one thing that they appear to not want to tell us is the benefits of masks, because they know as soon as they do that, everybody's going to go and hoard masks. But it's more and more slowly coming out that the recommendation is that even a cloth mask or something like that when you're outside or when you're shopping or doing those things can be beneficial both when you're sick and also to reduce the likelihood of infection as well. So again, we're dealing with these chronic stresses and we don't affect them physically, and they last for a longer period of time. And so that can have more of an impact on our kidney and our kidney function. All right. Questions on that? That is the beauty of the sympathetic mechanism for how it controls our glomerular filtration rate. And I will go ahead, since I did so much writing here, I will save that whiteboard. And again, these are things that I'll post on our Canvas site for us. All righty, let me see where we are. So there's that. So let's go back to our screen. So now, as we talked about our sympathetic nervous system, adjust the afferent and efferent arterioles, constricting those to, again, maintain a higher pressure, even when less blood is going to the kidneys. And when it stimulates the afferent arteriole, again, it's stimulating the smooth muscle cells, which are those granule cells. So when the granule cells are stimulated by the sympathetic nervous system, not only does it cause them to constrict, which is going to increase the pressure, but the other thing it does is it causes them to release their hormones and their hormone-like proteins. So the other type of extrinsic control are hormones. Luckily, 2 of these are hormones we've already talked about. Our antidiuretic hormone we already talked about in the endocrine system. Our atrial natriuretic peptide we've already talked about twice. We talked about it in the cardiovascular system, and then we talked about it again in the endocrine system. And we hinted at this Renin-Angiotensin-Aldosterone system. We did talk about aldosterone, but we'll talk about this in a little bit more detail now as well. Now, it's a teeny bit early for a break, but this is a good natural stopping point. So let's go ahead and take our second break here. And then we come back from our second break, we will talk about our hormonal regulation of glomerular filtration rate. I told you we'd learn everything you ever wanted to know and more about glomerular filtration rate today. So we're going to go ahead and do that. Let's go ahead and take a 10-minute break. So that means coming back at 10:10. And again, oops, now start the recording. All right, so any questions before we take our break? Excellent. So hopefully our intrinsic mechanisms, and there are 2 of them, make sense. We're halfway through our extrinsic mechanisms. There are 2 of those. We've done the neural one. We will do the hormonal one next. All right, I will go ahead and pause the recording now. We'll take a 10-minute break. In the recording, bring up people, bring up my chat window, and we are good to go. All right, excellent. So we have talked about 3 of our 4 mechanisms for controlling the rate of our glomerular filtration rate. And so the last one we need to talk about are hormones. And like I said, the good news is a lot of this stuff are things that we have already talked about. Starting first with our antidiuretic hormone. And notice I have 2 separate questions here. Where is antidiuretic hormone produced and where is antidiuretic hormone released? So humor me, where is antidiuretic hormone produced?
Posterior pituitary gland.
Nope. Where is it produced?
The kidneys.
No. You are absolutely correct that antidiuretic hormone is released by the posterior pituitary. So absolutely it is released by the posterior pituitary. But if we remember back to our endocrine system, the posterior pituitary was unique in our endocrine system because it was basically the only endocrine, quote unquote, gland that does not produce its own hormones. Absolutely, it is produced by the hypothalamus. So it is the hypothalamus where it is produced, but you are absolutely corrected in that it is the posterior pituitary where it is released. Excellent. Now, what did our antidiuretic hormone do again? Anyone remember? Antidiuretic in simple terms?
Hold on to fluids.
Yeah, hold on to water, absolutely. It told the kidney, remember we said the target was the kidney, and it told the kidney to hold on. Now we get to be a little bit more specific. It is going to increase our reabsorption of water. Remember, the goal of that is that we get an increase in the volume of the blood, and that leads to an increase in our blood pressure. So we talked about that when we did that. Although now we can talk a little bit more about how that is going to occur. It is going to occur first in our distal convoluted tubule and in the principal cells of our collecting duct. And the way it does it is by, and I'll write it up here so it's out of the way, it controls, oops, if I spell it right, the expression of a special protein called an aquaporin. An aquaporin is a special protein that, when expressed on the surface of the cell, allows the cell to reabsorb water. One of the interesting things about the distal convoluted tubule and the collecting duct is that normally those cells are impermeable to water. Water cannot get through them. So the only way we can reabsorb water is by expressing those aquaporins. When we express those aquaporins in those regions, it allows us to absorb more water. That is going to increase our blood volume, increase our blood pressure. And the net result of that for the urinary system is it's going to concentrate the urine. It draws more water out of the filtrate back into the blood. And so the urine we produce is more concentrated. Remember, our distal convoluted tubule and our collecting duct is where we do the fine tuning. And this is where we're doing our fine tuning, where we're telling our kidney, hey, we need more water. Our blood pressure is low. Our blood volume is low. Bring that back. And then that's also how we talked about if you suppress antidiuretic hormone, like with caffeine, like with alcohol, you produce and release less of that antidiuretic hormone. And so basically your kidneys bring back less water. So you produce more urine, it's more dilute urine. Your blood volume goes down, your blood pressure goes down, and that drop in blood volume, drop in blood pressure, that dehydration can be some of the factors that produce that hangover you have the next day. Or like when you have that, quad shot from Dutch Brothers, although I don't know if you're allowed to go to Dutch Brothers anymore, so you make a quad shot at home. And when you drink that coffee, you have to pee as a result of it. So that is their antidiuretic hormone. All right. The other one we've already talked about is the atrial natriuretic peptide. And of course, where is that both produced and released?
Atrium.
Yeah, the atria of the heart, absolutely. In the atria of the heart, we produce it. Again, remember, this is in response to stretch of the atria, right, from an increase in blood pressure. And so when that occurs, that says our blood pressure is too high. We need to get rid of some of that extra volume of blood. So we do that in a couple ways. We are going to, excuse me, stop reabsorbing, tell the kidney to stop reabsorbing sodium, because of course where stuff goes, water follows. So if more sodium is released in our urine, more water is going to stay out there in it. Atrial natriuretic peptide suppresses our antidiuretic hormone and aldosterone, which we'll talk about in a second. And again, antidiuretic hormone brings back water, brings up the blood volume and pressure. So this will bring it down. And it increases our water loss. So it brings our blood pressure down, which when it's too high. And that's what that stretch of the atria. Because again, remember, when the blood reaches the atria, it is supposed to be at a blood pressure of 0 millimeters of mercury. So if it's above that, that's a problem. So again, no real new information there. Well, a little bit. We talked about these aquaporins now. That is definitely something that is new. But let's get to the system we haven't talked too much about yet. And this is this Renin-Angiotensin-Aldosterone system. We did talk a little bit about renin. Renin is a hormone-like protein. And I'm sure I say it here, but I'll say it again up here. And that hormone-like protein is basically released from the kidney when there is a drop in blood pressure, a drop in blood volume. Obviously, there's a massive amount of blood coming to our kidneys, so the kidneys is a perfect place to be monitoring the condition of the blood. And so if there's a drop in blood volume or blood pressure, then what happens as a result of that is those granular cells, those juxtaglomerular cells around the afferent arteriole release renin. And it's this hormone-like protein. One of the things that renin does, one of the primary things the renin does, is it activates a protein called angiotensin 1. Angiotensin 1 is produced in the liver. However, what's interesting about it, and let's see what I have written here. There you go, excellent. What renin actually does is it activates an enzyme, and that enzyme is actually located in the lungs. And that enzyme that is located in the lungs, which again, if you think about it, kind of makes sense because it's another location where there's a large amount of blood going to. And what that enzyme in the lung does is it converts the angiotensin 1 into angiotensin 2. And angiotensin 2 is our hormone. This is our hormone. This is the one that is going to have a bunch of major elaborate effects on our body. We'll talk about and identify those in just a second. Whoops. Oh, and we'll do it right there. Thought I had more on the screen. All right, excellent. So let's talk about the things that angiotensin 2 does. Angiotensin 2 is our hormone, and it has a large amount of effects. So let's go through them and think about how this would affect our blood pressure. Because remember, the trigger, let's go back to all of this. Oops, not that. The trigger was a drop in blood pressure. And we know that can be a bad thing because if blood pressure goes down, that could lead to a decrease in our glomerular filtration rate. Oops. And that's a bad thing. So what we need to do is we need to bring up that blood pressure. We need to increase that glomerular filtration rate. So let's talk about the effects of angiotensin 2. The first thing it does is it constricts arterioles. And notice the key word there being everywhere. What I mean by that is, of course, both in the kidneys, as we just talked about, if you constrict the afferent and efferent arterioles, that helps to bring the blood pressure up, congests it. So afferent arteriole and efferent arteriole. And we just finished talking about with our sympathetic nervous system how that will help to maintain our glomerular filtration rate. But the other thing that it's going to do is it is going to cause the constriction of our systemic arterioles as well. And if we constrict a bunch of systemic arterioles, we're going to increase, right, the systemic blood pressure as well. So that's going to bring up our blood pressure in general, not just the kidneys, but it will bring up our blood pressure everywhere. Angiotensin II stimulates, again, the kidneys to reabsorb more sodium, more chloride, right? So more stuff. And that stuff is primarily ions. But as we know, where stuff goes, water follows. So it's going to lead to the reabsorption of more water. And of course, if more water comes back into the blood, our blood volume goes up and our blood pressure is going to go up. But there are going to be other big effects of angiotensin II as well. One of the things it does is cause the release of aldosterone from the adrenal cortex. And again, this is going to require some reminding for me. In what zone of the adrenal cortex is aldosterone released from? So I know you guys learned that endocrine stuff and were thinking you would never have to deal with it again. And yet here we are. You got a 1 in 3 chance of getting it right. Which of our zones of the adrenal cortex is responsible for releasing aldosterone?
Zona vesicularis.
Oops, I lost it. It is, well, so let's think of it this way. Maybe this will help. So we need to know the zone. The other thing we need is to think of it in terms of aldosterone is a specific hormone, right? Aldosterone. And what class of hormones is aldosterone the most common one of?
Androgens.
No, it's that, well, no, it's a mineral corticoid. Excellent. It's the mineral corticoids. And as we see here, the zone it is released from is the zona glomerulosa. Excellent. So again, it's produced in the zona glomerulosa. It is a mineral corticoid helping us to deal with our electrolytes. And of course, our most common electrolyte was what again?
Sodium.
There you go. It is sodium. So our aldosterone helps us to regulate the reabsorption of sodium. So by producing more aldosterone from the zona glomerulosa of the adrenal cortex, that is going to, oops, I didn't have that there, so I'll write it here. We're going to reabsorb more sodium, and as we've talked about, where sodium goes, water is going to follow, so that will increase the blood volume, increase the blood pressure. Angiotensin II also stimulates the hypothalamus. The hypothalamus, remember, is our Illuminati control center of the endocrine system. But again, it's part of the nervous system as well, and as we've talked about, it controls a lot of other functions of the body. It plays a role in regulating heart rate and things along those lines as well. And in our hypothalamus, we actually have a thirst center. So angiotensin II stimulates your thirst center. And when you stimulate your thirst center, guess what you do?
Drink water.
There There you go. You get thirsty, and when you get thirsty, you drink, increasing the amount of water that's in our digestive system, increasing the absorption of water from our digestive system, which increases blood volume, which increases blood pressure. And as if all of that's not enough, angiotensin II also stimulates the release of antidiuretic hormone, which again causes those aquaporins to be expressed, causing us to draw back in more water. Notice the goal of all of these are the same. We get more water into the blood vessels. And if we get more water into the blood vessels, our blood volume goes up. But more importantly than that, our blood pressure goes up. And notice this is an extrinsic system. The goal immediately for the urinary system with a higher blood pressure, we're going to maintain a better glomerular filtration rate. But it really causes a systemic increase in blood pressure as well. We've got all this here on the board here with all of my writing in between. We've made a nice, beautiful mess of this. But if you look at the pretty picture from your textbook, I'm going to clear the drawings to be able to do that, we can see that your book's got this great flow chart that shows all of this process. So notice, again, we have our homeostatic imbalance. Our glomerular filtration rate has gone down, right, because of a decrease in blood flow to the kidney. That decrease in the blood flow to the kidney, that means we have a decreased filtration pressure, right, urine production. But luckily we have those juxtaglomerular cells, those granular cells that are able to identify that, and they release renin, that hormone-like protein. That hormone-like protein stimulates the formation of angiotensin in the liver, but more importantly, it leads to the activation of that angiotensin into the hormone angiotensin II. And again, remember that occurs in the lungs. Right, so the activation occurs in the lungs, whereas the production occurs in the liver. And that angiotensin II is, as we can see, a dynamic hormone with a lot of effects. It causes the constriction of the kidneys, arterioles, right, which increases glomerular pressure. It leads to the stimulation, get that out of my way, of systemic, where is it? Here we go. Systemic constriction of arterioles, which are going to increase the blood pressure. It causes the release of aldosterone, which leads to sodium reabsorption. Right? It causes sodium and chloride and potassium to be reabsorbed. And again, when that happens, water follows. Antidiuretic hormone causes, again, the reabsorption of water, bringing all of that water in, whether we bring solutes in or water directly, we're increasing blood volume. More blood volume, constriction of blood vessels leads to an increase in pressure, and that increase in pressure, both locally and systemically, brings the glomerular pressure back up, and we get our normal glomerular filtration rate. So we have this pretty picture that kind of sums it all up and puts it all together. And we also have this pretty flow chart that shows all of our mechanisms as well. It shows our neural controls, our hormonal controls, right? These two out here are our extrinsic controls. And these two are the intrinsic, right? Our myogenic mechanism and the juxtaglomerular apparatus, which does that tubular glomerular mechanism. So here we see all four of our mechanisms working together. In this case, when blood pressure drops, how we can bring it back up so we can maintain that appropriate glomerular filtration rate. All right, and just that easily, we have now identified and described all of the fun things, everything you ever wanted to know and more about our glomerular filtration rate and what's happening in our renal corpuscle. All right, and let's go back one more time. Questions on any of that? All right, if we are satisfied with the first step in this process, then what we can do now is we can start talking about steps 2 and 3. Again, it is important to remember that both steps 2 and 3 are occurring at the same time. Tubular reabsorption and tubular secretion occur together. And it's also important to remember that the majority of both of these occur in the proximal convoluted tubule. All right, so maybe we'll even write that on the top here to remind us of that. All right, both occur at the same time. And both, well, let's say it this way. The majority of both processes occur in the proximal convoluted tubule. Excellent. So we get that reminder of that. Then we can do that. The other important thing to remember is that they're going to use similar processes, similar mechanisms. Really, the only difference is the direction. Our tubular reabsorption is from the filtrate to the blood, whereas secretion is from the blood to the filtrate. Or again, we went medium to medium, we could do structure to structure. Reabsorption is from the tubule to the peritubular capillary. Secretion is from the peritubular capillary to the tubule. So again, either of those are good ways of describing this. Oops, and now that I've written that there, I need to move it. So let's get this out of the way. And again, go back one slide. Here. So the direction is different. And obviously the substances that are moved are different as well. Like we talked about, it is primarily good things we want to bring back in. Our ions, our metabolites, and other types of electrolytes and things along those lines. The things we want secreted are waste materials, toxins, or other chemicals that are produced by the body, medicines that we take, things along those lines. However, you will notice there is some interesting things. Notice potassium is on both lists. Calcium is on both lists. And stuff like that, because as we've talked about, remember our blood is basically continuous with our interstitial fluid. And in our interstitial fluid, we want to have a low level of potassium. So we want to make sure we have enough, but we don't want to have too much. And as we've also talked about, calcium makes cells do wonky things. So definitely, not only can we have too little calcium in the blood, we can also have too much calcium in the blood. So since those things are very tightly regulated, it's not surprising that they will both be reabsorbed and secreted. In fact, the movement of these is much, much less important than, for instance, and let's change the colors, as we'll see, the reabsorption of sodium and chloride, both of which there are high levels of in the blood, are going to be so much more important. And we're not going to worry as much about potassium and calcium because we're going to want to make sure that we have appropriate levels, but it's not about too much. We want high levels of sodium, high levels of chloride. So we're going to want a lot of those in, and so we're going to move a lot of those back. All right. And like I said, we are going to reabsorb over 99% of our filtrate. We produce close to 200 liters of filtrate during the course of the day, but you only produce 1 to 2 liters of urine. So the vast majority of 99% of the filtrate is going to be reabsorbed. And as I talked about, both reabsorption and secretion, the majority of that occurs in the proximal convoluted tubule. So for instance, 70% overall of the reabsorption happens in the proximal convoluted tubule. However, for instance, when we talk about things like glucose, amino acids, proteins, vitamins, things along this line, these pretty much 100% of these things are going to be reabsorbed in our proximal convoluted tubule. And so when we talk about these two processes, again, reabsorption and secretion, we're going to talk about separately. but they are occurring at the same time, and the majority of them is happening in the proximal convoluted tubule. So let's go ahead and go to the next slide to talk about the processes, the filtrate and the formation of the urine. Again, movement of fluid and solutes between the blood vessels and the tubules. Methods, plural, for moving them. These take us all the way back to 430 when we were talking about our movement mechanisms. We are going to use some passive transports. Remember that involves our filtration formation, which we've already talked about. But we can also talk about simple diffusion and facilitated diffusion. Can someone remind me again what the difference between simple diffusion and facilitated diffusion is again? What does simple diffusion mean?
High concentration to low concentration.
True, all diffusion is going to be a high concentration to low concentration. So that's going to be true for both simple and facilitated. So absolutely, we're talking about diffusion. So you're absolutely right. We're going from a high concentration to a low concentration. But what's the big difference between simple and facilitated?
Use the channels.
There you go, absolutely. With simple diffusion, remember, there is no channels or no proteins, let's say it that way. With simple diffusion, there is no proteins. Basically, all it is, there's my arrow, there it is, a movement of the substances across the plasma membrane without any proteins that assist in the process. With facilitated is going to use some types of proteins. Those proteins can be channels or they can be carriers. Remember, the big difference between a channel and a carrier is that channels do not change shape, carriers do. If you think back to let's do that instead. If you think back to 430, we talked about channels and carriers, and if you don't remember that, then I strongly encourage you to go back and look at that when we talked about the cell and we talked about the plasma membrane. We talked about these things, and the key to it is a channel basically is like a door frame, right? It can be gated, it can be open and closed, it can be locked, it can be selective, right? Limiting how things come through size-wise and stuff along those lines, but it doesn't change shape. A carrier is like that revolving door, but not like one at a fancy hotel where they have it electrically set up and monitored. And again, the example I always like to use is the zoo. If you've ever been to the Sacramento Zoo, if you're going to leave the Sacramento Zoo, there is a big, huge metal revolving gate that you have to use to get out, and you have to push on that gate to change the shape of it for it to come through. And that's what carriers do. So again, if you don't remember these transport mechanisms, both the active and passive, I strongly encourage you to go back to chapter 3 where it talks about the cell, and make sure you read about these membrane transport mechanisms, because they are going to be important moving forward as we talk about these things. So facilitated diffusion means it's using a protein, some type of channel or carrier to allow things in, where simple diffusion is straight through. And of course, as we've also talked about, osmosis is just a fancy word for this simple diffusion of water. So again, these are going to be the types of passive mechanisms that we are going to use for doing that. And I need to go ahead and clear that, because while there are plenty of passive transport mechanisms that we're going to talk about, as we also talked about, we want to be able to be selective. Oh, actually, I should talk about that here. As was mentioned, when we're dealing with diffusion, we are dealing with concentration gradients, and we are also dealing with osmolarity gradients. Remember, osmolarity is the measure of the amount of stuff. It doesn't matter what that stuff is. And remember, as we talked about, salt, when you put it in water, becomes two things, sodium and chloride, whereas glucose, even though it's a much larger molecule, when you put it in water, is still just one thing. So remember that salt water has a higher osmolarity than that glucose water does, because it's just a matter of counting the stuff and not caring what that stuff is. But remember, the key with reabsorption and secretion is we want it to be selective. And so if we want it to be selective, we're going to need to use active transport for that. We're going to need to use pumps, we're going to need to use exchangers and co-transporters and antiporters and all the fun things that go around with that. And again, I know those are terms that may be tricky for some people because you may not remember it. I don't have enough room here at the bottom, so I guess let's cheat and go back to our whiteboard so we can do a quick reminder of what those things are, because we do need to make sure we understand that going forward. Again, we are going to need a plasma membrane, and there's one there. And when we talk about a pump, remember basically what we are talking about is some type of protein that is in the plasma membrane, and its job is to use ATP. Oops. ATP to basically move a substance the direction we want it. So for instance, if we have a sodium pump, or let's actually talk about here, this is a great, move that there. If I move that there, I can remind us of the most common pump we have in our cells, and that is our sodium potassium ATPase, or what is also known as the sodium potassium pump. Sodium potassium pumps use ATP to move what two ions?
Sodium and potassium.
There you go, sodium and potassium. And which way does it move the sodium?
In? No,
Well, again,
out.
yeah, remember with a pump, we typically, if we're using ATP, we're using it to move things the way they don't want to go. Sodium wants to go in the cell. Remember, there's a high level of sodium outside here. And let's remind ourselves of that. High level of sodium. High amount of sodium inside the cell. And a low amount of sodium outside. High amount of sodium outside the cell, so this is our outside. And this is our inside. And here we have a low level of sodium. Excellent. So sodium wants to come in normally. So here, if we're using the ATP, what we have to do is we are moving sodium out. And how many sodium do we pump out again?
3.
3. Excellent. 3 sodium are going to be pumped out thanks to that ATP. And that, of course, means that potassium is going to be moved in. And how many potassium does it bring in again?
2.
2 potassium. Excellent. So again, that is our most common pump. In fact, if you have a cell at rest, close to 25% of the ATP that is being used by that cell, when the cell is just sitting there at rest and not doing anything else, is being used to activate these sodium potassium pumps, just to maintain those concentration gradients. Right? This is an example, if you remember, of what we called a primary active transporter. Right? This is where we directly use ATP to do work. This is important because if there are primary active transporters, that means there are also secondary active transporters. And these indirectly use ATP to do their work. And the two classic examples we had for this, let me a second to move these things out of my way and get to do some more drawing. Are our symporters and our antiporters. Remember, with these are both actually, let's go ahead and do this. Both of these are what we call co-transporters because they move two molecules. Oops. But the key to this is they use the energy of one. So they're going to move two molecules, but they use the energy of one to do the work for us. And there are two types, symporters and antiporters. Notice our sodium-potassium ATPase moves two things as well. But again, remember, it's directly using ATP. Remember also, it doesn't have to use two things. Another example of one of these pumps that we talked about was, for instance, like a calcium pump. Where we directly use ATP, I keep not abbreviating, I'm not capitalizing, to move calcium out of the cell. So again, pumps don't have to move two things like the sodium potassium ATPase, but co-transporters do. In this case, what we do is we mentioned sodium really wants to come inside of a cell. So what we can do is we can say, okay, sodium, you can come in. We let that sodium come in. But the energy from that sodium coming in, we use to do work. If you remember, we used the example when we were back in 430 of like the water wheel. Right? Back in ancient times, before you could plug your grinder into the wall, what you had to do if you wanted to grind your flour is you built your flour mill right next to a river. And what you would do is you would put this big, huge paddle out into the river. And as the water of the river flowed by, it would move that paddle. And you were able to use the energy of the water going by to do work, to grind your flour or to mine your gold or do any of those types of things. In this case, we let sodium come in. And with a symporter, it brings something else in as well. So in this case, for instance, it could be used to bring in an amino acid or a glucose or something like that, right? That is our symporter. An antiporter starts the same way. It allows sodium to come in. All right, sodium, you really want into this cell. That's fine. I will let you into the cell, but I'm going to use your energy of coming into the cell to kick something out. And that something could be, for instance, like a hydrogen ion or maybe a calcium, because we know those do pesky things. So I can use the energy of the sodium to do work. Symporter bringing it the same direction, antiporter going in opposite directions. Notice nowhere in this drawing have I put an ATP. So the question becomes, how is this an active process? And it comes back to this key being indirectly. Notice there's a large amount of sodium outside and a little bit inside. But if we keep letting sodium come in, then there's going to be less outside, more inside, and eventually sodium reaches equilibrium. And if sodium reaches equilibrium, it can't do work for us anymore. So what happens is, as soon as that sodium comes inside, what we're going to do is kick it back out again. As soon as that sodium comes inside, we are going to kick it back out again. So notice over here, we're using the ATP. So we're not directly using the ATP, it's indirect use of the ATP. But if I don't use ATP to kick that sodium out, these transporters, these co-transporters won't work. So this is our secondary active transport. This is our indirect. And again, we are going to use both of these mechanisms. We are going to use both active and primary and secondary active transporters in the movement of our substances. All right, let's go back to our screen. Excellent. So both the pumps, the exchangers, co-transporters, antiporters, all of these things that we're going to be using to move these things in and out. And again, as we just talked about, directly or indirectly ATP, and while your body's at rest, about 25%, 20 to 25% of your oxygen is being used by your kidneys while you're just sitting here quietly, which makes sense. About 25% of the blood is going to your kidneys, so it makes sense that's where 25% of the oxygen is going. Those numbers work out. All righty, here we have the wall of our proximal convoluted tubule. So oops, I lost my annotation and I lost you people and my chat window. Every time I change, everything goes away. All right, excellent. So again, this is our proximal convoluted tubule. That's what we're focusing on here. And this would be the wall of that. We see the nice prominent villi of them here. Here's our peritubular capillary right here. Here would be our lumen where our filtrate is located. And as you notice from these illustrations, there are going to be two main routes that a solute can take, water and our solute can take, to get back into the capillary. Now, these cells, if you remember back again to chapter 3, have cell junctions that combine them together. And these have tight junctions. These tight junctions, remember, are interlocking globular proteins that are embedded in the plasma membrane, and they do two things. They limit the movement of water between the cells. But the other thing they do is they form a barrier. One of the things, remember, we talked about is our plasma membrane is a fluid mosaic. A protein or a channel or a receptor that's right here on the plasma membrane isn't going to stay right there. It has that liquid quality. And again, we use the example, if you have a glass of water and you pour a droplet of oil on top of it, that oil is going to spread over the surface of the water and it's going to look still. But as we know, all the molecules of fat are going to be continuously floating around. And so any protein here is going to be floating around as well, and it has the ability to move. Well, what we don't want is, for instance, a sodium transporter out here on the apical side to get to the basal or the lateral side. So by having these tight junctions, it also helps to separate the two membranes so that the transporters that are located on the apical surface are going to be different from the transporters that are located on the basal surface. And that's going to be important for our function. All right. Now, as I mentioned, there are two ways to get through. Those tight junctions limit what can get through, but it doesn't stop it. So water and solutes dissolved in water do still have the ability to sneak between the cells to work their way back in. Water, and in some cases, as much as 50% of some ions, chloride is an example of this, can actually sneak in paracellularly by sneaking in between the walls of the cells. However, the majority of our solutes are going to have to pass through a cell. And when they pass through a cell, that is our transcellular reabsorption, where it has to go through, if you think about it, it has to pass through two membranes. It has to pass through the apical membrane and it has to pass through the basal membrane. And because of those tight junctions, typically the channel or the protein that assists its movement from the apical side is going to be different from that on the basal side. All right. So two ways for things to get through. They can go through the cells, and if you go through the cells, you have to pass two membranes, or you can sneak between the cells. Those are going to be the ways that we're going to get stuff out of the filtrate, out of the lumen of our tubule, and back into our blood. All right. Now, when we're processing our filtrate and forming the urine, let me clear that. One of the important things to remember, especially when we're talking about our transmembrane or transcellular transport, is that most of it, because it is active, most of it is going to use some type of protein, some type of carrier, right? Some type of specific protein that is going to bring it through. Now, typically they're going to move in one direction. So glucose into the cell on the apical surface, sodium into the cell on the apical surface, and things along those lines. But the important thing to remember is that not only are they going to be different on the apical and basal surfaces, but they may be different types of transporters as well. So again, if we think of in simple terms of a cell, let's go ahead and draw our cell here. And again, for simplicity's sake, we will put some microvilli on this side just so we can tell the apical surface from the basal. If we think about reabsorption pathways, notice in this case we would want glucose to enter the cell on the apical surface, but on the basal surface, we're going to want that glucose to leave the cell. So again, and how we bring it in and how we bring it out are typically going to use different types of proteins, different types of mechanisms. So we have to cross those two membranes, often in different directions, often using different mechanisms, different type of transporters. The other thing to remember, and it looks like I'm not going to have enough room, so I'll have to go ahead and clear that picture, is that these transporters have a maximum rate. Again, the example I like to think of this is in terms of like if you're at the mall. Again, back in ancient times, people used to be able to go to the mall, and there was a great story of a situation where BTS did a surprise performance at a mall, and they were up on the second floor of the mall. And so there was this mass movement of people trying to get upstairs to see BTS when they were active. And again, with stairs, where you are actively moving up, you're using the energy to move up. There's still a limit of how many people can get on that stairs. But where they really had the problem with the escalators. Escalators would be example of active transporters. The mall is using the energy to move people, but you're still limited on how many people you can get on there and how fast they can move them. And the escalators became so overloaded that the escalators actually broke as people were trying to get upstairs to go see BTS. Which again, makes no sense to me. It's not like they're the Jonas Brothers or anything, but it's some people's cup of tea and that works. So that's the point. How many things can be transported in a certain amount of time is what we call the transport maximum. And this transport maximum is an important number because basically it is going to determine if a solute is in the urine. How much of that solute you have and what your transport maximum is going to determine whether or not that substance is going to be in the urine, right? Oops, no, wrong button. Right? Glucose, for instance, it's 180 milligrams per deciliter. Amino acids are 65 milligrams per deciliter. So let's think about this. If in your filtrate, you have 40 milligrams per deciliter of amino acids, is that number less than our transport maximum or more than your transport maximum? Is 40 greater than 65 or less than 65?
Less than.
There you go. So are we going to be able to get all of the amino acids on my escalators and get them out of the filtrate?
Yep.
Absolutely. Right. And notice glucose has a very high. right? What is a normal, again, I know we had a couple groups that did, talked about glucose levels in the blood, including the ones that did diabetes. Anyone know what a normal healthy level of glucose in your blood would be?
80 to 120.
Excellent, I like that. Glucose levels should be about 80 to 120 milligrams per deciliter in your blood. Is that number less than or greater than your transport maximum?
Less than.
There you go. So what that means is we have enough escalators that in normal blood glucose levels, all of that blood glucose is going to be reabsorbed. However, as we talked about with uncontrolled diabetes mellitus, what can happen is glucose levels can get as high as 400 milligrams per deciliter. And if you have 400 milligrams of deciliter of glucose in your blood, do we have enough escalators to get all of that back into our blood?
Nope.
No, and so as a result of that, glucose is going to be present in the urine, right? As we talked about in ancient times, that's one of the reasons why doctors get paid so much. Not only because they have to deal with COVID victims, but because in ancient times, if you had a baby that was crying, one of the things they would do is taste the urine to see if it was sweet, if there was glucose present, because that would be an indication that that child had diabetes. So the transport maximum, if I give you a transport maximum and I give you how much of something there is in our filtrate, you should be able to tell me whether or not it's going to be present in the urine. Okay? It's just a real simple, which number is higher kind of thing. Right? And here again, just to beat that dead horse, here we see that example. Again, glucose reabsorption is occurring here. It's being brought in. Notice happens to be brought in with a co-transporter of sodium. So it's brought in on the active on the apical side, then leaves on the basal side. And we'll actually talk about these mechanisms in just a minute. In a normal, healthy individual, 100% of our glucose is reabsorbed. But if someone has diabetes mellitus, then the level of glucose is far greater, and it's not all going to be reabsorbed. I stole this illustration, and of course, that case of having glucose in your urine is called glucosuria. But I love this animation. Another instructor did this, and so I stole it from them. But what we can see here, oh, come on. So again, notice at this point we have far more. Can you guys see that? Is that moving? Okay, excellent. Far more glucose in our filtrate because of that uncontrolled diabetes. And so only a little bit of it is transported. And so a lot of this is being filtered out instead in our urine. One of the other things too, if you think about it, if you have more glucose in your blood, if you have more glucose in your filtrate, more stuff in your filtrate, what likes to follow stuff?
Water.
Water. So that's what's going to happen. Water is actually going to follow it as well. So typically when someone has uncontrolled diabetes, not only do they have glucose in their urine, but they typically produce more urine, a higher volume of urine, and that urine tends to be more dilute because of that excess amount. And it can lead to dehydration for those individuals as well. All right. So determining how much of something gets back is going to be determined by its transport mechanism. Every single solute has a transport maximum that determines how much of it gets brought back. And when you weigh how much is in the filtrate by what the transport maximum is, you can determine what percentage, 100% or less than 100%, of that solute is going to be reabsorbed and whether or not it's going to be present in the blood. All right, questions on that? All right, we didn't get quite as far as I wanted to, but I think I've been talking for a long time, and although I'd like to start this process, I worry it's going to take us beyond, and I've talked already for too much, so we're a little behind. We do have a lot to talk about then. That means on Tuesday before our, on Monday before our Wednesday exam, but again, you have a week and a half now to start looking at this material. Make sure you look at the rest of the lecture notes. I encourage you to read the textbook for this part of this to make sure that you understand it, put all the pieces together. And so, like I said, Monday should mostly be review. So we will start with the 5 major processes that take place in the proximal convoluted tubule, and we'll pick up there. So I think this is a good natural stopping point. So any questions on any of this before we call it a day? All righty then. So with that, as always, I'll stick around for a few minutes if anybody has any last minute questions or concerns or comments. Otherwise, please be healthy. Please be safe. Please take care of yourselves and your families. Enjoy your spring break as much as you can, but also study hard, work hard. And again, please, between now and Friday, make sure you take that practice lecture exam. So, or it's really the practice lab exam, because that'll be the format of the lab exam. Make sure you take that to make sure that your system is compatible. And if it's not, like I said, the good news is you have a couple weeks to try to resolve that, because the test is 2 weeks from today. But let me know if you have any problems, and we'll try to see what we can do to work that out and help you. If you need resources, the ARC has Chromebooks and as laptops that they were providing for students who needed those resources. I know they were doing that at the beginning of the process. I don't know if they still have more of those, but you can contact the school and contact the library to see if that is the case, if those are things that can be checked out, or other things that you can do. So please let me know. Please take that so we know that this works, so we can minimize the number of problems we have on the actual exam itself. So like I said, be safe, have a good vacation, and I will see you. Like I said, I'll be around. I'm not going anywhere. I'll be working on the exams. So please email me if you have questions. I can meet you on Zoom. We can chat by email. I have office hours next week, so I'll be around. In all likelihood, you will be around. So be safe, be healthy, have fun, and I will see you next Monday. All right, I'm going to stop the recording now.